Things like genetic variety and the way things operate in the ecosystem are preserved through conservation biology, and it supports the populations and species. Molecular genetic approaches may be used to identify persons, animals, populations, and conservation units, among other things. This is beneficial for more than simply tracking and conserving genetic variation. The study of genetics is crucial in various areas of conservation biology research because it was once a minor part of conservation study, but it is now a key priority. Researchers investigated the worth of genetic information and discovered that environmental or demographic variables may be more significant. Most scientists use species to quantify biodiversity. Species are used to assess biodiversity value through gathering lists of protected species, identifying endangered species within domains, and forming the basis of biosecurity policy. Then the discovery of hybrid zones and mixed population, which can be caused by introgressive hybridization between closely related species, can have serious effects on conservation, politics, and the economy. Lastly, wildlife disease monitoring can follow domestic and wild animal morbidity and mortality, determine changes in disease patterns over time, and discover disease outbreaks earlier.
Identification of individuals, species, population, and conservation units. First, identification step. Identification steps start with isolation of samples. Samples might be feather, blood, skin, and etc. Next, the sample will identify using PCR. In PCR, molecular marker will include within the sample taken. It can reveal some characteristic about the sample. It will amplify by PCR, then the PCR product will undergo DNA sequence in order to know what species and population are there. Molecular markers have three types: protein, mitochondrial DNA, nuclear DNA. First other type of protein is allozyme. Allozyme are protein products of genes that are encoded by a single gene locus. Since they represent genes of known function, they are considered to be type 1 markers. Allozyme have been used to assess population structure. Second, mitochondrial 12SR DNA is highly conserved and has been applied to understand the genetic diversity of high categorical levels such as in phyla. On the other hand, the 16SR DNA is often used for studies at middle categorical levels such as in families or genera. Third, mitochondrial protein coding genes, due to their faster evolutionary rates compared to ribosomal RNA genes, the mitochondrial protein coding genes are regarded as powerful marker for genetic diversity analysis at lower categorical levels, including families, genera, and species. RAPD markers, random amplified polymorphic DNA (RAPD) markers are analyzed by using PCR to amplify the segments of nuclear DNA. The use of a single primer, usually 8 to 10 base pair long, that attach to both strands of DNA and low analytic temperature increase the likelihood of amplifying multiple regions representing a particular locus. AFL3 markers is amplified fragment length polymorphism. Is a multi-locus technique that involves translation, digestion, and PCR amplification. Macrosatellite marker are multiple copies of short tandem repeats, generally 1 to 5 base pair long, located in both coding and non-coding regions and fairly evenly distributed throughout the eukaryotic genomes. Macrosatellites are codominant markers with biallelic or multi-allelic presentation in an individual or population respectively.
Use of molecular marker in defining species and subspecies. Hybridization. What are hybrid zones? Although the use of the phrase hybrid zone infers that hybridization, the genetic blending of dissimilar groups occur inside a zone. This also suggests a specific geographic area. Thus, from a spatial perspective, hybrid zone comprise of set of clines or gradient in trait values or allele frequencies. The main goal of speciation research is to comprehend how hybridization affects interspecific divergence. How do hybrid zones arise and how do they change? Although they can occur in situ, hybrid zones are typically thought to result through the secondary contact of diverging lineage. Hybrid zone could be relatively old and persist long enough for some or all genome composition in hybrids to approach an equilibrium between migration of allele and selection among them.
Molecular ecology has now become an important part of wildlife disease management. Rapid detection of even low intensities of viral, bacterial, and parasitic infections is now possible using tests based on polymerase chain reaction or PCR. PCR is a technique that amplifies or makes more copies of specific DNA regions to confirm the presence of pathogens. Currently, PCR-based diagnostic tests allow for the swift detection of a number of diseases in wildlife such as the Syazana forest disease, Nipah, tuberculosis and malaria, all of which are directly responsible for endangering wildlife. Here I will show you how we can detect the presence of pathogen by using PCR method. I will take Nipah virus as an example. Instead of using conventional PCR for the detection of Nipah virus, we will use real-time PCR because it has been shown to be 1,000 times more sensitive as conventional PCR. Next, the procedure for real-time RT-PCR is started by taking the sample from bodily fluid of the infected animals. The sample then undergoes several chemical reactions to obtain a mix of the infected animal's genetic materials and the viral RNA if present. If the viral RNA is present, it will be converted to DNA by reverse transcriptase enzyme and it will be amplified in the thermocycler. If the viral DNA is present, the fluorescence level is high due to the DNA was dyed with fluorescence materials.
As a conclusion, biodiversity mainly refers on variety of life existent living on Earth. The main cause of changes on biodiversity are due to pollution, invasive species overexploitation and climate change. Therefore, rapid gain of detailed information on population at risk allow better understanding in order for effective recommendation in decision makers. Advances in molecular techniques such as DNA sequencing, molecular markers, and PCR techniques can be used for screening, characterization, and evaluation in conservation of genetic diversity. That's all from us. Thank you so much for watching.